Welcome to A-Minder, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello, and welcome to the June 2020 edition of A-Minder. This is Anusha, and today we'll be covering two sections under the main theme of treatment and testing, but with a focus on preclinical testing, which is mostly done on animal models for Alzheimer's disease. So first, we'll cover 11 papers that look at multi-targeted approaches and neuroprotective compounds. And then we'll turn our attention to 10 papers that target specifically improving memory, cognition, and or behavioral alterations in Alzheimer's disease. So without further ado, let's get started. This first paper is called Long-Term Oral Melatonin Alleviates Memory Deficits, Reduces Amyloid Beta Deposition Associated with Downregulation of BACE1 and Mitophagy in APP-PS1 Transgenic Mice. By first and last authors Sun and Song, respectively. This was published in Neuroscience Letters and investigates how long-term oral melatonin affects A-beta deposits and levels, spatial learning and memory, APP processing, mitochondrial morphology and turnover, and they do this all in an AD mouse model, the APP-PS1 mouse. They report that long-term oral administration of melatonin improves the normally impaired performance in the Morris water maze. So in other words, that the spatial and learning memory deficit in their AD mouse can be improved after oral administration of melatonin. Authors also found reduced deposition of A-beta, reduced damage to the mitochondria, reduction in expression levels for markers of mitophagy, as well as APP-related proteins. So I really hope this holds true when they extend the research to people. Next up, a paper titled Berberine Nanoencapsulation Attenuates Hallmarks of Scopolamine-Induced Alzheimer's-like Disease in Rats, published in Current Clinical Pharmacology by first author Saleh and last author Abdel Monaim. Authors in this paper looked at a compound, an isoquinoline alkaloid called berberine, that has multiple pharmacological actions, including the inhibition of both A-beta production and acetylcholinesterase activity. But its poor absorption, bioavailability, and uptake in the brain reduces its pharmacological benefits. So they wanted to improve this compound's efficacy by encapsulating it in nanoparticles. So today we're going to talk about the effects of these nanoparticles on scopolamine-induced cognitive impairments in the Wistar rat. They had so many groups here to compare. So uninduced rats, so they were not treated with scopolamine. And they were either given saline, polyethylene glycol, or the vehicle for berberine, which is chitosan. And then they had an induced rat group that were untreated, or an induced rat group, induced with scopolamine again, treated with either berberine nanoparticles, or the positive control donepezil. They found that nanoparticles, in comparison to berberine or donepezil, significantly improved performance on the Morris water maze, inhibited acetylcholinesterase and A-beta 42 formation. This happened at a sixth of the dose of non-nano berberine, so for a small particle, it was no small feat. Up next, from authors Zengin, Montesanto, and others, a paper in the journal Foods titled Chemical Characterization and Bioactive Properties of Different Extracts from Fabigia clypeata, an Unexplored Plant Food. Authors looked to the plant Fabigia clypeata, MEDIC, to see if its extracts could inhibit enzymatic activity that could improve Alzheimer's disease state, as well as type 2 diabetes and skin hyperpigmentation. They found that when tested in the cell lines FADU, which one of these cell lines was actually a cancer cell line, and SCC25 cell line, that F-clipieta, when extracted into methanol, inhibited the activity of acetylcholinesterase, butyrylcholinesterase, tyrosinase, and alpha-glucosidase, and showed antioxidant potential, meaning that it could also benefit type 2 diabetes, as well as Alzheimer's disease, cancer, and skin hyperpigmentation. Within the extracts, they identified derivatives of caffeic acid O-hexoside and ferulic acid, which may be the active ingredients at play here. Certainly more has to be done here to test what it can do in a more physiological setting, but it's promising nonetheless. Now, a paper titled Exploration of Multi-Target Effects of 3-Benzoyl 5-hydroxychromin 2,1 in Alzheimer's Disease Cell and Mouse Models. First author is Lin, last author is Chen, and the journal is Aging Cell. Authors here looked at lidocalcone A, which is a natural aromatic ketone, and 5 synthetic derivatives for its effects on tau hyperphosphorylation and oxidative stress. They used human cells to express delta K280 tau RD DS-RED and found that lidocalcone A and the derivative of that compound, LM031, reduced tau misfolding, reactive oxygen species, improved neurite outgrowth, and reduced caspase-3 activity in 293 and SHSY5Y cells expressing this delta K280 tau. Mechanistically, they showed LM031 upregulates HSPB1 chaperone proteins, NRF2, and Krebs signaling pathways to inhibit apoptosis and enhance neuronal survival. I haven't read the paper, but I appreciate how much information they provided, even in the abstract. So things are looking good for the paper. Next, we have mulberry fruit extract alleviates cognitive impairment by promoting the clearance of amyloid beta and inhibiting neuroinflammation in Alzheimer's disease mice. This is a paper by Liu, Du, and others published in Neurochemical Research. As you probably guessed from the title, authors here looked at the efficacy of mulberry fruit extract in improving AD-like cognitive impairments in APP-PS1 mice. Daily oral administration improved performance on spatial learning and memory tasks like the Morris-Water maze, which, spoiler alert, you'll see a lot of in this section. They found also reduced A-beta plaques and neuronal cell loss in the cortex and hippocampus of treated mice, as well as elevated anti-inflammatory cytokine IL-4 and reduced pro-inflammatory cytokines including IL-6, 1-beta, and TNF-alpha. So that was a really straightforward abstract to get through. So we'll move on to the next paper, which is actually our halfway point for this section. So this paper is called Fish Oil and Primrose Oil Suppresses the Progression of Alzheimer's-like Disease Induced by Aluminum in Rats by Al-Akbi, Ali, and others in the Journal of Oleoscience. Here, rats were divided into 5 groups. 1 control normal, 1 control AD-like, which they didn't go into detail in the abstract or really in the paper as to what they meant by AD-like. But they also had 3 other groups, and these 3 groups were fed with either primrose oil, fish oil, or a mixture of both oils. Gas chromatography showed that primrose oil and the mixture of both oils contained gamma-linoleic acids, eicosapentaenoic, and dicosahexaenoic. So compared to all of the diets, the mixture of the two reduced elevated erythrocyte sedimentation rate, plasma butyrylcholinesterase, brain malondialdehyde and nitric oxide, reduced brain histopathology, and all of this to the greatest extent to all of the diets tested. It also increased good cholesterols, or HDLs. What this means for AD symptoms seems like a logical next step to take after this paper. So moving on now to the Journal of Agriculture and Food Chemistry, we find ourselves looking at a paper called Torula rodin from Sporadiabolus pererosaeus attenuates D-galactose ALCL3-induced cognitive impairment, oxidative stress, and neuroinflammation via the NRF2 NF-kappa B pathway. By first and last authors Zhang and Qian. The compound terularodin, a carotenoid pigment, is a potent antioxidant and was used here to test the effects on cognitive impairment in the D-gal ALCL3-induced ICR mice, which is another model of AD. The results indicate that terularodin relieves cognitive impairments and A-beta accumulation, reduces glial overactivation, and lowered inflammatory cytokines. Western blot analysis suggested that this was through the activation of NRF2 translocation and inactivation of NF-kappa B both in vivo and in vitro. They suggested that the NRF2 translocation thereby upregulated HO1, which ultimately contributed to the results that they found. I guess eat your veggies, people. That's what you should take away from this paper. Next up, a paper by El Said and Ghoniam et al. From Oxidative Medicine and Cellular Longevity, which is, to me, a very, very specific journal. So the title here is Antia, a natural antioxidant product, attenuates cognitive dysfunction in streptozotocin-induced mouse model of sporadic Alzheimer's disease by targeting the amyloidogenic, inflammatory, autophagy, and oxidative stress pathways. Authors used sporadic AD induced in mice to investigate the effects of Antia, which is found in certain mushrooms, yams, and berries. Streptozotocin was injected ICV into mice to induce AD, and then Antia was given IP for 21 days. They found that it improved performance in the Morris water maze and that it had a protective effect on GSH, MDA, NF-kappa B, IL-6, and TNF-alpha, as well as A-beta. Your homework is to read the paper to interpret what they mean by protective effect on a protein. But they also suggest that this protective effect is through the JAK2 STAT3 pathway, thereby reducing neuroinflammation. This next paper is titled Astra agrotides Turks extract attenuates Alzheimer's disease-associated cognitive deficits and vascular dementia-associated neuronal death. This was published in Anatomy and Cell Biology by Zhang, Han, and others. They tested the antioxidant potential of Astra agrotides extract, AAE, for therapeutic effects in rat models of AD and vascular dementia. In addition to the in vitro evidence of attenuated neuronal excitotoxicity with AAE, they showed that administration of AAE for 7 days orally improved performance on behavioral assays of memory following scopolamine administration. AAE also attenuated induced microglial activation and hippocampal neuronal death after 2-vessel occlusion and hypovolemia. So another potential therapy for dementia. Next, by Zhang, and others, including a third Zhang, published in the Journal of Biomedical Reports, a paper titled Total Flavonoids of Selaginella pulvinata Alleviates Cognitive Impairment in Mice. Authors tested total flavonoids of Selaginella pulvinata, which they shortened to TFSP, which has been shown to have neuroprotective and antioxidant effects on scopolamine-induced mice to test its effects on memory impairment. They showed that TFSP improved memory in behavioral testing, as well as increased blood acetylcholine levels and reduced oxidative stress in the brain, which is good news not only for AD, but also Parkinson's and dementia research as well. Our final paper in this section is called Exploiting Citrus Aurantium Seeds and Their Secondary Metabolites in the Management of Alzheimer's Disease, published in Toxicology Reports by first and last authors Baker and Gengehi, respectively. I usually paraphrase, but I really wanted to share with you the way that they phrased this particular sentence because I just think it's so beautifully written. This study looks at the byproducts in fruits, which they term, ready? Nature's golden gift for human health. Isn't that amazing? So, back to my words now. They specifically looked at the seeds from Citrus aurantium to study neuroprotective effects on rats given ALCL3 for 3 weeks. Treatments with donepezil, hesperidine, and limonoids were administered 2 weeks prior to ALCL3 induction. They found significant reduction in cognitive deterioration as assessed by Y-Maze performance. High dose of limonoids and both doses of hesperidine significantly increased ambulation frequency when compared to the positive controls, so that's the donepezil, in open field tests. Low dose of limonoids and both doses of hesperidine also protected from changes in ACE and A-beta levels. So they suggest that the compounds in Citrus aurantium seeds, which are usually wasted in huge amounts, could be recycled to provide prophylaxis for AD. And I'm all for reducing waste while improving health. So that was a good note to end on for that section. Let's switch gears slightly to looking at the changes to cognition, memory, and behavior that are related to AD that can be targeted and improved on. The first paper we'll be covering is from the Journal of Chemical Neuroanatomy. This is called Crocin Attenuates the Granular Cells Damages on the Dentate Gyrus and Pyramidal Neurons in the CA3 regions of the hippocampus and frontal cortex in the rat model of Alzheimer's disease. The first author is Hadipour and the last author is Hatef. As loss of synapses and neurons in cortical and hippocampal brain regions are a hallmark of AD, the authors studied the effects of a neuromodulatory compound, crocin, which is a major component in saffron, on these brain regions in an AD rat model. They found that loss of cells and synapses in the cortex and hippocampus of AD brains could be reduced within 12 days of crocin administration. Next, we have a paper titled Protease Nexin-1 Protects Against Alzheimer's Disease by Regulating the Sonic Hedgehog Signaling Pathway. This was published in the International Journal of Neuroscience by authors Li Xi et al., aimed to explore the function of the protease Nexin-1 in the sonic hedgehog pathway as it related to Alzheimer's disease. They injected Nexin-1 into APP-PS1 transgenic mice alongside wild-type controls, which they followed up by inhibiting Nexin-1 or sonic hedgehog, and then tested the effects of this on hippocampal cell survival, learning, and spatial memory. They determined through these pharmacological means that Nexin-1, through its inhibition of a sonic hedgehog, can reduce hippocampal cell death and improve spatial learning and memory. Let's turn now to a paper published by Chen Li et al. In Fundamental Clinical Pharmacology, titled Mineralocorticoid Receptor Antagonists Mediated Cognitive Improvement in a Mouse Model of Alzheimer's Type, Possible Involvement of BDNF, H2, SNRF2 Signaling. Amyloid beta was injected into mice to induce cognitive impairment and were then tested for memory using the Morris-Water maze test. Spironolactone and eplerenone, two mineralocorticoid receptor blockers, were administered for 14 days post-induction. Expression of neuropeptides were assayed and the Morris-Water maze tests were repeated. They found that mineralocorticoid receptor inhibition attenuated the cognitive decline induced by A-beta injection, potentially by elevating levels of the neuropeptide BDNF in the frontal cortex and hippocampus, which may serve as a neuroprotectant. Next, a paper titled PNU 282987 alleviates A-beta-induced anxiety and depressive-like behaviors through upregulation of alpha-7 nicotinic acetylcholine receptor, which they've shortened to alpha-7 NACHR, by ERK serotonin receptors pathway. By first and last authors Chang and Qian, respectively, this was published in Neuroscience Letters. The authors set out to better understand the role of alpha-7 NACHR on A-beta-induced mood disturbances as well as on the regulation of MAP kinase pathways. Mice were injected with A-beta oligomers and tested for anxiety and depression-like behaviors using elevated plus mazes, open field, and forced swim tests. Mice which had alpha-7 and ACHR displayed far less anxiety and depressive-like behaviors, and pharmacological inhibition of the ERK pathway had the same effects as alpha-7 and ACHR activation on reversing the changes in serotonin receptor levels affected by A-beta induction, giving further insight into the mechanism underlying the improvements seen with alpha-7 and ACHR activation. So now we're at the halfway point for this section. Up next, from Journal of Alzheimer's Disease, we have, by authors Liu, Zhu, and others, muscone ameliorates synaptic dysfunction and cognitive deficits in APP-PS1 mice. Muscone, or muscone, forgive me if I'm mispronouncing this, is a neuroprotective compound that has been effective in treating cerebral ischemia, but the authors wanted to explore its effects on the APP-PS1 AD model. Novel object recognition and Morris water maze tests were used to observe behavioral phenotypes that could be targeted with muscone or muscone. That's the last time. No, there's one more time I say this word. So synaptic morphology, plasticity, cell viability, and A-beta were measured. Hippocampal plasticity and memory impairment normally observed with these mice was attenuated using muscone, which they posit works by increasing degradation of histone deacetylase 2. So this could be yet another potential therapeutic for Alzheimer's disease. Next, we have Icarine improves cognitive deficits by reducing the deposition of beta amyloid peptide and inhibition of neurons apoptosis in SAMP8 mice by Wu Luo et al., published in Neuropotective. Wanting to test a traditional Chinese herb derivative, Icarine, for its potential to treat and prevent AD, this group used the SAMP8 AD mouse model to observe the effects of intragastric administration of Icarine. They found Icarine reversed the learning and memory impairment in the Morris water maze while downregulating BACE1 protein expression and ultimately A-beta 142 expression through Western blot analysis. They also found a reduction in the pro-apoptotic protein expression and increased the anti-apoptotic protein expression, supporting the idea that Icarine could be used to treat AD. The next paper we'll be exploring that addresses cognitive changes in AD models is one titled Dehydroefusol Rescues Amyloid Beta 25 to 35 Induced Spatial Working Memory Deficits by Fukuda Takeda et al. In Plant Foods and Human Nutrition. First, authors induced AD-like cognitive decline using A-beta 25 to 35 in mice. After initial testing of learning and memory using the Y-maze, mice were injected with dehydroefusol, which they deemed effective in rescuing the cognitive decline they initially induced. That was short and sweet. Next up, we have evodimine derivatives improve cognitive abilities in APP in brackets SWE, PS1 in brackets delta E9 transgenic mouse model of Alzheimer's disease. Published in Animal Models in Experimental Medicine. The first author is Pang, the last author is Zhang. And these authors, among other researchers, looked at the compound evodimine, which is a quinoline alkaloid. It's already been shown that evodimine improves learning and memory, but it's also quite cytotoxic. So they were looking at its derivative in its effects on improving learning and memory, but perhaps without the cytotoxicity associated with the original evodimine compound. First, cytotoxicity was assayed using the WST8 test in SHSY5Y cells. Then APP-PS1 mice were given either evodimine or its derivative before being tested in the Morris water maze. They found several oxo and thio derivatives that performed better than evodimine with reduced cytotoxicity, but that's all I'll say about this, so you'll have to visit the paper for specifics. Kodagale, Taksande, and others published our next paper in neurotoxicology. This paper is titled Involvement of Hippocampal Agmatine in Beta 1 to 42 Amyloid Induced Memory Impairment in Neuroinflammation and BDNF Signaling Disruption in Mice. They wanted to test the effects of agmatine, which is a biological amine, and it has a potential for therapeutic benefit in neurodegenerative disorders. So they looked at how the impairment in cognitive abilities induced by A-beta-1 injection could be improved with agmatine administration into the hippocampus. They saw prevention of memory impairment in radial arm maze performance and novel object recognition performance, but did not see alterations in APP and BACE, as in BACE, expression levels. They suppose these improvements may be a result of reducing neuroinflammation. Our final paper is also brought to you by first and last authors Kodagale and Taksande, respectively. They published this paper in Pharmacology, Biochemistry, and Behavior, and it's a paper titled Agmatine Reverses Memory Deficits Induced by A-beta-1 to 42 Peptide in Mice, a key role of imidazoline receptors. In this paper, they explored what agmatine, again a biogenic amine, does to A-beta-142 induced memory deficits in mice. Mice were given a single ICV injection of A-beta-142 and evaluated using the Morris-Water maze for learning and memory. Researchers also assessed hippocampal A-beta-142, TNF-alpha, BDNF, and IL-6 immunocontent and found that increased levels of these proteins as well as cognitive impairment was rescued with IP daily injection of agmatine and imidazoline I1 and 2 receptor agonists. However, antagonists to imidazoline receptors attenuated the effects of agmatine on cognitive impairment and hippocampal protein levels. The authors suggest, therefore, agmatine and imidazoline receptor agonists could provide therapeutic relief for individuals with AD. That's it for me today. I hope there was at least one paper in here that captured your attention. And until we meet again, goodbye.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMinder. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMinder team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.